Good day, this is Professor Will, CBMD PhD, and today's Wednesday, May 11th, 2022. It is 6:45 and 12 seconds, a.m. Eastern Standard Time. Today's research, mosquito brains. So let's hop to it here. How mosquito brains encode human filter so they can seek us out. Date May 4, 2022. Source, Princeton University. And some strains of Aedes aegypti, the mosquito that carries Zika, malaria, and dengue fever, have evolved to bite humans almost exclusively. Mosquitoes bane of backyard picnics and deadly in Zika and dengue-prone regions. Most of the world's mosquitoes are opportunistic, willing to drink blood from any nearby source, but in some regions, the mosquitoes that carry Zika, dengue, and yellow fever, Aedes aegypti, have evolved to bite humans almost exclusively. But two succeed as a specialized feature, depending on just one species, ours. To survive, they must have evolved incredibly precise targeting strategies. How do they do it? Researchers set out to try to understand how these mosquitoes distinguish human and animal filter, both in terms of what it is about, human filter, and what they cue in on, and what part of their brain allows them to cue in on those signals. The researchers have discovered both parts of the equation. What is it the mosquitoes are detecting, and how do they detect it? Researchers sort of dove into the brain of the mosquito and asked, what can you smell? What lights up your brain? What is activating your neurons? And how is your brain activated differently when you smell human odor versus animal odor? Imaging mosquito brains at very high resolution to watch how the mosquito identifies its next victim. And to do that, researchers had to first genetically engineer mosquitoes whose brains lit up when active. And then the team had to deliver human and animal-flavored hair in ways that the mosquitoes could detect well inside the team's custom-built imaging equipment. Human odor is composed of dozens of different compounds, and those same compounds, in slightly different ratios, are present in most mammal odors. None of those compounds is attractive to mosquitoes by itself, so the challenge was to determine the exact blend of components that mosquitoes use to recognize human odor. The team concluded that two chemicals, decanol and undecanol, are enriched in human odor. They patented a blend featuring decanol that they hope can lead to bait-attracting mosquitoes to lethal traps or repellents that interrupt the signal. Researchers collect hair, fur, and wool samples, and for this research, the team used odor from 16 humans, 2 rats, 2 guinea pigs, 2 quail, 1 sheep, and 4 dogs. For the human samples, researchers had a bunch of great volunteers. Researchers had them not shower for a few days, then stripped down, naked and lie down in a Teflon bag. Why naked? Because cotton and polyester and other clothing fibers have their own smells that would distort the data. And once they conquered the technical challenges, retrieving the human and animal odors non-destructively, designing a system that allowed them to puff human odor at the mosquitoes in the imaging setup, creating a wind tunnel to test simple blends or single compounds, and breeding viable strains of mosquitoes whose brains respond to the equipment. They began gathering data. Before this study, researchers speculated that mosquito brains must have a complicated, sophisticated technique for distinguishing humans from other animals. Quite the opposite, it turned out. The simplicity surprised the researchers, despite the complexity of human odor, and the fact that it doesn't really have any kind of human-specific compounds in it. The mosquitoes have evolved a surprisingly simple mechanism for recognizing us. And if researchers created a statistical test to differentiate human odor, it would be very complex. But the mosquito does something remarkably simple, and simple usually works pretty well when it comes to evolution. In other words, simple solutions Tends to be true over evolutionary time. Mosquito brains have 60 nerve centers, called glomeruli, singular glomerulus. The team had hypothesized that many, maybe even most, would be involved in helping these human-dependent mosquitoes find their favorite food. When researchers first saw the brain activity, they couldn't believe it. Just 2 glomeruli were involved. That contradicted everything they expected, so they repeated the experiment several times with more humans, more animals. They just couldn't believe it. It is so simple. Of the 2 nerve centers, one responds to many smells, including human odor, essentially saying, hey look, there's something interesting nearby. You should check out. While the others respond to only humans, having to may help the mosquitoes home in on their targets. One of the biggest eureka moments in the project. Researchers worked for a couple years to get the transgenic mosquitoes that they needed, and then they found that they didn't have a good way to deliver human odor. And so researchers worked for another year or 2, coming up with ideas to try to figure out how to deliver enough human odor in a controlled enough way to see a response. Then the first time they tried this new technology, they described in the paper the new way of delivering odors. They actually saw a brain respond. It was incredible. By determining the glomeruli that mosquitoes use to detect humans and identifying what it is they are detecting, decadal and undecanal, the team has an eloquently straightforward answer to their questions. And if this were purely a neuroimaging paper, there would be some questions remaining. And if this were purely an odor analysis paper, there would still be unanswered questions. A purely behavioral paper, same thing. But one real strength of this project is that they were able to bring in so many different methods and the expertise of so many people. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.